So, Rodger, let me start off directly. I think CRISPR has been claimed as like the discovery of the 21st century. Do you believe that?
Well, first of all, hi guys. Thanks for the invitation. I don't know. I certainly think that CRISPR, or the CRISPR-Cas system, I should say, has huge potential. We know already that in many areas outside human therapeutics, it has changed the industry, certainly in the ag bio environment. I think for human therapeutics, it will take some time, but I also believe that it will have a huge impact on how we see certain diseases. I'm by training a pediatrician and a geneticist, so I certainly believe that there's a huge potential, no question.
Okay, that's great. And I'm always surprised by how many companies are rushing into the space. I mean, just like Editas, Intelia, Caribou, just founded very lately and raising money. What's your view on that?
I think there's a very limited number of companies, and I don't expect too many companies to get into that place simply because of the intellectual property situation. I think very much in difference to many other fields. The IP, despite some of the hype and some of the misinformation we have in the media, is I would say is not totally clear. That's very obvious. But I think you would have and you will have access to some of the so-called foundational IP, be it in biotech or be it in pharma. Pharma does understand this really well, and so I don't expect too many more companies. Serious companies coming up in the human therapeutic field simply because they will not find investors backing them, because it is known by now that there is this IP they need to get access to.
Yeah. Actually, about IP, I had a question as well. I mean, I know the fight between like the Broad was Zhang and Church against Doudna, but I never read so much about the name of Charpentier in that fight. Like, how are you placing in that? Yeah?
Well, I think that's a tricky one. Yeah, that's something. So how to put that best? So Church is not relevant in this setting. He's at Harvard, but the Broad Institute is one of the players. Actually, the person most relevant for us, but probably the field, is Charpentier, because when she invented, she was in Sweden as a professor. There's professor's privilege, and she does own the IP as an individual. So it means that she is the owner. She's not only an inventor, she's the owner. I'm very happy about this because I think she all deserves this, because she was a driving force behind the field. Actually, if I may make a few comments, which I think, I don't know who is in the audience, but I think it's interesting to realize. So Emmanuelle is a scientist, and actually she worked on the basic mechanism of how actually bacteria fight viruses. So a very basic question. She was working on the group A Streptococcus as a model organism in the lab, and group A Streptococcus is actually characterized by having a lot of phages, superantigens, and so that was a question. And so now a basic science finding was actually converted into what I, coming back to your first question, personally believe is probably one of the biggest, if not the biggest, invention in the modern biotech world, certainly in my view, to be named close to or next to PCR or whatever it is. So it's a very interesting for me personally that this comes out of basic science. And the person behind that already in 2010 was Emmanuelle, with no doubt. And I think when you look at the number of awards she has received compared to others in the field, in particular to Fang, I think she's at 35 and Fang is 3. Academia fully recognizes that. And it's just a different style of a different individual, not being grown or not being at the West Coast, but being a European, in that case, French person who focuses more on her science than on media hype.
I guess only the Nobel is missing now.
I'm not sitting on the committee. She's now a member of the Royal Academy of Sweden, so maybe she has more insight into that. Actually, probably not for herself. Yeah.
Actually, a funny fact. I mean, from the last Nobel Prize award, we wrote an article. We thought CRISPR would probably win it. And actually, we wrote like CRISPR, you'll have to wait 1 more year, and it's probably one of the most read articles on the website anyway. It's pretty funny. And I was wondering how you started with Emmanuel, on how you met, how you started the company. That's fun.
Yeah, actually, we met in the mid-90s, so I'm already a pretty old fart. So I was at Rockefeller in New York as a postdoc, and Emmanuel was a postdoc as well in the same lab. And so that's how paths crossed. And I've told the story many times, but I think still it's an interesting story. She called me, I think it was early 2011, and I was at Sanofi at the time. I was the global head anti-infectives for Sanofi, and she said, "What do you think about CRISPR?" And she has a very nice French accent, so I said, "What are you talking about? CRISPR?" Yeah, so I didn't really know. I had heard about CRISPR because she was just working on a Nature paper, but I did share the data she shared with me, and I got the okay to do that with our gene technology group, and people were really thrilled. They got it right away that this could be a big thing. Not so much for human therapeutics, I have to say, but more actually for manipulation of cells, whatever it is. And so we looked at that, but the time was not there yet, I would think, in particular with an organization like Sanofi, and it probably would have been with any other big company. And so over time, the idea came up that we may want to build a company. And a very good friend, he's a VC, was a former board member of a company of mine. I knew that he had time, and I said, "Can you have a look?" That's Sean Foy, the other founder. And he said, "Sure." And he came back 4 weeks later, and he said, "You need to leave your job." And I said, "Sure." But actually, I did about a year later, and we founded CRISPR Therapeutics, having access to this IP, and then started to build a company actually in Basel, Switzerland. So none of us is Swiss. But we thought, there are probably 3 locations at the time in Europe where you potentially could do something like that. It was Basel, Zurich, certainly Cambridge, Oxford, and we were kind of hoping for maybe Munich, Berlin, or whatever, but we came to the conclusion probably not the right time for that. And then we got first financing from Versant Ventures with 25 million in a Series A. But it was very clear very early on that this is not the right way to go. And so over the course of 2014, we decided then to extend the investor base and actually then move operations to Cambridge, Massachusetts. And we did that in 2015. Early on, after JP Morgan, those people who are in the field know there was a crazy JP Morgan, so extremely easy to access money, in particular if you were called CRISPR. We raised another 69 million, or whatever, 64, I think we had an 89 million, and actually built out operations in Cambridge. And a year later, I have to say, it's just a year now, we increased from 7 people to almost 80 now. We closed 2 very significant deals. So we have a cash position in the company itself of 250 million. We have a cash position in a JV with Bayer of 300 million plus. And so we are very happy where we are, but I do think it's a very unusual story, and it has to do with CRISPR and the interest of the field and the CRISPR technology.
Yeah, you took all my questions now. You're almost competing with Moderna on the cash now. I think they have still a bit more.
Yeah, I think it's a very different business model.
Yeah.
But I think one of the reasons we and others are so interested in CRISPR, it works. I think thousands of academic labs use the technology and with that drive innovation. So that's very, very good. And that certainly has an impact on how you build a company, what's your corporate or business strategy. And so, I do think it's a very, very special situation, and probably not comparable to other companies out there. But yeah, that's where we are.
And I'm curious about why. I mean, on the location, it's pretty interesting because you started in Basel and then you moved to Cambridge, where already all the other CRISPR companies are basically, and where the research is also done. Like, what's the reason, or the main reason that it exists?
Yeah, it's investors. I don't know. So in parts, yeah. So I think it's a big question for Europe. If I may advocate this here as a European, actually, and it's actually sad to see that we can build big biotech companies, in particular at the East Coast, but also at the West Coast. We do have fantastic examples in Europe for some very few top-notch biotech companies, but unfortunately it's a very small number compared to what we see in particular right now in Boston. The reasons, there are different reasons. Certainly one of the reasons is access to capital. The VC environment is very different. If you want to access capital markets, if you want to interact with investment bankers, you name it, analysts, it's so much easier in the US. New York is just next door. Then access to talent and a very different mindset in part with researchers. I think this cuts both ways. So when you go in Boston and want to recruit, let's say, a top-notch academic, the first thing the guy asks for is a cap table. So means what kind of ownership do I have in the company? When we do this in Europe, you have to explain people what a cap table is. I find this much better. But fact is, there's a very different mentality, and I'm a little sad about the fact that we were not in the position to really build out a company like CRISPR in Europe. Maybe we do something in the future, but right now we are primarily at the East Coast.
Because Versant also have operation in Basel, so I guess maybe that's linked also to Basel initially.
Yeah, well, Versant is one out of many investors by now. We're very happy to have Versant in the very beginning, but the investor base is extended significantly, and with that, obviously, the impact of individual investors ceases, and that's a good thing.
All right, and back to the few companies active in the space, do you think the approaches will be complementary on developing CRISPR therapeutics on the longer term, or will it be kind of winner takes all?
No, I don't think. I think what you have to keep in mind, and I think that's an important. I don't know if the comment is important, but I personally think it is important. When you think about big pharma, most big pharma companies have maybe 4 or 5 disease areas they play in, max. And when you think about CRISPR, the applicability of the technology is certainly not only in monogenetic diseases, but actually throughout any kind of disease area you can imagine. So I think the idea of many people, including analysts, that you're competing around a small area is doesn't make a lot of sense. So that's in a longer way to answer your question. I think there's enough room for a number of companies. I think we don't have to be complementary. I also do think, as a CEO of a company, that if you look too much what's happening right and left, you certainly will not be in a position to focus on what you should focus on, and that's development of your drugs. There's a lot of hype out there. There's a lot of expectation out there, and a lot of distraction. And so I think we are well advised to make sure that we get it right. And if companies like Intellia and Editas get it right, perfect. But in the end, I don't care.
Great. And now, move more to the clinics and how recent the technology is, on a general point of view. The technology is so recent, and seems like you have some off targets and other thing, unwanted thing, uncontrolled. How is that affecting into clinics?
Well, so obviously the key value driver for anything we do in the industry is products, and you only have products at least if you have a chance to move something into clinical development. Again, personally, I do believe that the challenges of the technology are only in parts linked to gene tox, so with that to off-target. The bigger challenges we have, and that's very similar to what we have heard in gene therapy, in parts is manufacturing, not so much of the CRISPR-Cas components, but if you are, for example, applying cellular-based medicines and gene edit, then for sure you have to look into details of process development. So that's something to address. Having said this, I think that is addressable with all the challenges we are aware of in the industry. I think you might want to divide the work world for CRISPR in two areas: ex vivo, and that's really cellular-based medicines, be it stem cell manipulation or whatever it is, with the huge advantage that you actually can qualify your drug product outside the body. So very happy about that we have that opportunity. Here, the challenge is less gene tox or off-target, but more so pharmacology. So how many cells do I have to edit, and how many cells actually do I need to bring back to have my therapeutic approach? And that's very different depending on the disease. And if you go in vivo, I think we have a very different challenge. Delivery, everybody talks about delivery. That's true in parts, depending on the organ system. Maybe less so for the liver, certainly for muscle, certainly for the brain. And there, for sure, safety considerations are very, very different. And the bar with the regulators, that's our expectation, is higher. But again, I think the in vivo applications with regard to clinical development, in my view, are trailing maximum by a year or 1.5 years, considering what we expect for ex vivo applications when they enter the clinics.
And what are you focusing on?
So I obviously can only talk about what's on the website. But I think it's relatively clear. We have done a deal with Vertex end of last year that disclosed hemoglobinopathies as a so co-development and co-commercialization. We have disclosed cystic fibrosis together with Vertex. We have done actually next door a pretty big deal with Bayer in a JV also last year that does include additional hematology applications, hemophilia, PID, so primary immunodeficiency, the iron parts, and cardiovascular, something certainly more for the future.
Great. I think we have time now for questions. Question around there, if you can bring a mic.
Thanks. Rodger, when can we see the first in man with CRISPR-Cas9, edited product?
Yeah, we always get that question, and it's a very, very important question. So I'm a bit careful. I personally believe that we might see the first clinical trials coming out of academia. So we are aware of a number of later stage preclinical development programs in academia. For the industry, biotech, be it with the partners or standalone, I think reasonably something in early 18, if things go okay.
Thank you.
What are you going to do in cancer?
So cancer is, and I unfortunately didn't have a chance to listen to too much of what's going on today, but at least the last panel I listened to a bit. So I can't tell you what we do in cancer, but I can tell you what you could do in cancer. So I think one of the interesting aspects, and I disagree a little bit that if you go to an investor and talk about immuno-oncology, that they're now tired of that. I think the field is just opening up. In particular, when you think about gene editing, what we see is that the potential to multiplex, so to go after 4 or 5 targets at the very same time with the CRISPR-Cas system in T cells, for example, it doesn't have to be only T cells, can be NK cells, whatever, is huge. So it's a very significant potential to go into allo. So to move actually from autologous approaches to allo, something Selectis is trying to do. Though the CRISPR-Cas system has huge advantages over talents when you want to go after more than 2 targets. And so I think that is something one can look at. Editas and Intellia did early deals. We have talked to, or actually companies talk to us, pretty much all of them, and we're not interested in a deal right now.
Any other question? I have one more on the on your next plan. I mean, the only successful IPO this year, kind of like, the last one was Intellia, who just announced the amount over 100 million. Editas did as well, over 100 million. Do you think like it's, you think IPO is a way for CRISPR Therapeutics?
Well, as I said, we are extremely well financed, and we are not the market environment, as we all know, is very difficult. So we keep all options open, but it's not a path we have to go down.
Any other question? I think we have 5 minutes left.
Standing between here and drinks or whatever it is. Yeah.
Actually, we can get you a beer and drink a beer 5 minutes now. Very good. Maybe you can talk about the deals you did with Vertex. I think total was like 2.5 billion, like possible. And that's pretty massive, if I'm correct.
Yeah, but these are people in the field who know about biodollars know what to do with biodollars. So I think the important thing is, and I think that, again, I don't know what kind of audience we have here, but I think what was absolutely surprising is that a technology where we have zero proof of concept, except for some animal studies coming out of academia, that pharma, it's not only Vertex, I can tell you that probably 7, 8 pharma companies, if not more, would have done deals like that if that could, are willing actually to jump into a field like this. So, and what was important to us, to keep it very, very tight. This is the only licensing deal we have done. And so it's really a target deal. It's not a deal around diseases, as you can imagine, with a genetics background that you can, for a given disease, have different targets. So Vertex was willing to go down that path for a very limited number of targets. Was willing to pay 105, so 75 million cash, 30 million in equity. And then actually finance our research in these areas, plus pay up to 420 million in milestones. I have to say, very much in contrast, that's all public, so I can talk about this, to the Regeneron deal we have seen with Intellia later on. So we have very early milestones, pretty much no commercial milestones. The message here is not so much the size of the deal, whatever. The message is that there is probably no larger biotech or no larger pharma company out there anymore who have their own R&D who are not working with CRISPR. They all work with CRISPR in their labs. Because there are no issued patents around the foundational IP yet, there's an interference, and there will not be only one, there will be many. Some people keep their powder dry and they wait, but there is not a company out there not using CRISPR for anything and anything you can imagine. So it is a very powerful technology, not only for human therapeutics, but also for tools, for whatever it is. And again, I find it fascinating that this comes out of a small bacteria called group A streptococcus, though all bacteria, or most bacteria, have a similar system. But what we also know, and I think that's interesting, none of the bacteria we have seen, and these are hundreds, species who have Cas9, in none of them the Cas9 works as well as from group A strep. So it's pure coincidence that Emmanuel worked as a lab bug with group A strep. So I think that's something also to keep in mind.
That's pretty crazy how it has been really discovered. Was pretty much random. Yeah. One more question.
Yeah, maybe a question on the ethical cornerstones frameworks that are being developed as we speak. I know that Emmanuel is part of the committee that is hosted by the National Academy of Sciences and the Royal Society. How is the company affected by that? How is the company also becoming active in this matter? That's something that might interest me.
So we are not affected so far, and I don't think we will be affected, but very clearly, and I think you reference germline editing and other aspects actually. So the company has a position on that, so we don't think that germline editing at this stage in particular makes any sense. The reason why that is, there are two reasons. So one is we believe that the understanding of the technology is limited. That is very obvious. And so I think going into germline editing makes zero sense, not only from an ethical perspective, but I really mean from a business perspective. And then there's the ethical consideration, where we as a company have a very clear position that we don't think that this is appropriate at this time and actually also for quite some time in the future. We do believe that the society needs a discussion about this because we have seen it not only because of China. Everybody cites it always China, but there are very active academics and clinicians actually also in the US promoting the use of the CRISPR system or similar systems in germlines. So we will have that discussion, but I think us as companies, we should stick to what we are hopefully good at, and that's certainly not germline editing, in particular not at this stage. I think that's very important. And I know Emmanuel has a very similar position.
Great. One very quick one.
Yeah, how do you see the competitive landscape with the alternative to CRISPR as gene editing technologies such as talent? I mean, we know that Selectis is about to start a clinical trial, so they are maybe a few years away. I mean, ahead of you in terms of at least going into the clinics.
Yeah, Ed Lanfier is not here, so we had a little bit of go at that last week in New York. Quite honestly, so first of all, we all use the gene editing tools as a tool. So we call it now CRISPR, TALEN, zinc finger, meganuclease, or whatever is out there. In the end, we don't care. It has to work. That's one part of the answer. The other is that if you have a really, really good zinc finger, and I come to talents in a second, I think there's justification for that. There's no question. If you have your off-target, if you have your pharmacology and your manufacturing under control, there's no reason not to use a zinc finger. The big issue is, in my view, is that over time, I think that the selection of an appropriate zinc finger takes so much time compared to a CRISPR system. So we have high throughput screening, so we can test about 10,000 guys within 4 weeks in biological assays for on and off target. So there is no way to compete. So unless you have very, very specific indications and you have a very good IP position like in part Sangamo has, obviously, there's no, in my personal view, over time, no justification to continue to work on zinc fingers. That's a little different for talents, but again, I think talents, Selectis does a fantastic job. They know really what they are doing. By the way, like Sangamo, very happy that these companies are out there and help actually to move the field forward. But when it, for example, comes to multiplexing, so Andrea and his team has worked for years to get 2 talents out there who can actually, with whom we can do multiplexing, at least for 2 targets. We and others have data just with the postdoc in a lab or in a company where we can go after 4 or 5 targets at the same time with 80% efficiency when it just comes to knockdowns. Obviously, replacement is a little bit more complex. So, yeah. I think there will be always applications. There's no reason for Selectis to not change horses, but I think the future is not only CRISPR for sure, but I think the standard is set by a system like CRISPR.
Great. Thank you very much, Rodger. It's a cup for you. I'm really glad you came with sneakers as well. It's awesome.